Thank you very much for the opportunity of being here today. I'm really sorry that I cannot be there. I'm going to talk about the Brazilian experience of microbial inputs, replacing chemical fertilizers and contributing to soil health. I work at the EMBRAPA Soybean Center in southern Brazil together with my colleague Mariangela Hungria. Plants need nutrients. We are not talking about a forest in equilibrium where we have nutrient cycling. Furthermore, if you can see here, the genotypes that we have today, they require much more nutrients than the ancestral genotypes that we have. Therefore, large-scale farming requires fertilizers. And we do have large-scale farming in Brazil. We are the first exporters of many commodities, soybean, coffee, orange juice, we have chicken, beef, and this is very important because it represents 1/4 of the gross domestic product. Therefore, if we are doing every year a larger scale farming, we are also consuming every year more chemical fertilizers. But the fertilizers, they do have problems. First, the low efficiency use of bioplants. For example, under tropical conditions of Brazil, nitrogen no more than 50%. Phosphorus is retained in our soils. We just use 15% of all fertilizers that we use, potassium 60%. Chemical fertilizers are also implicated in contamination of water and of emission of greenhouse gas. For example, 1 kilogram of nitrogen, it's more than 10 kilograms of CO2 equivalent. Price, importation, it's just incredible that Brazil relies on agriculture, but we are importing 85% of all fertilizers that we use in agriculture. And then prices are very expensive because we have to pay with international money. And also chemical fertilizers, they have negative impacts on soil diversity. Then what can we propose? Do we have alternatives for these problems? Yes, plant growth promoting microorganisms that can partially or fully replacing chemical fertilizers. And I'm going to talk to you today about the experience that we have in Brazil, things that we have on the shelf in products that we call as inoculants and that in community European you also call as biofertilizers. Let's talk about phosphorus. Start with phosphorus. We have many microbes that have mechanisms that can increase the uptake or the availability of phosphorus by mechanisms as solubilization and uptake. And after 15 years of research, we selected 2 strains of bacillus that they have the mechanisms of solubilization of less soluble sources like rock phosphates that we have, and also synthesis of phytohormones that can increase root growth. This is very important because then increases the uptake of water and nutrients. And the first inoculant for phosphorus was released in Brazil in 2019. And it's incredible because in just this short period, it's already used in 6 million hectares for soybean, for maize, for sugarcane. Potassium the same, it's a very soluble way that we lose the potassium, and we developed an inoculant that was released 2 years ago, based mainly on solubilization of potassium rocks and also increasing root growth, and therefore taking more advantage of the fertilizer that we apply. Our leader is the most beautiful biological process related to microbes, that's the biological nitrogen fixation. A microbial process that appeared in Earth far before any plant was here. And the most The most advanced these microbes with the capacity of nitrogen fixation associate with plants in different degrees of specificity. And the evolution, the most evolved form is the symbiosis with legumes. And we have in Brazil a long time tradition on research of nitrogen fixation. 70 years of research, and today we have about 100 rhizobium strains that are elite strains selected by the researcher for about 100 legume species that include tropical and temperate grain legumes, forage, trees, green manure, and the market with the rhizobium inoculants in Brazil last crop season was about 110 million doses per year. Most of them for the soybean, because soybean is our most beautiful and successful story. Brazil is today the first world soybean producer. And I can guarantee to you that this is only because of the biological nitrogen fixation. Brazil has invested 70 years of research on biological nitrogen fixation in the soybean. It's an exotic plant in Brazil. We did not have the strains. We imported the strains from US and Australia and started to see how they developed here under Brazilian conditions with our genotypes. And this was continued research. And if you ask me, I have 40 years of research in Brazil. If you ask me the limit of these strains, I will say, I don't know, because it's incredible. They were able to fulfill all needs of genotypes in the 1960s that had the capacity of producing 1,000 kilograms per hectare. And they continue to fulfill all the nitrogen need by plants today with the capacity of producing 6,000, 8,000 kilograms per hectare. But it's amazing because we are also pioneer in a study where while in other countries they said, well, once you introduce one strain, you can never improve that. No, we did more than 300 experiments in Brazil from north to south, and we saw that every year by adding fresh cells at sowing time of the soybean, we can guarantee in soils that hadn't received heavy inoculation before, but we can guarantee by adding fresh cells an increase in grain yield of average increase of 8%, and that's a lot. And then what's the importance of this type of research, believing in biological products? If you see USA, here a paper in a very prestigious journal with very prestigious universities. They did studies and said that to have an increase of 10% in grain yield, and this yield was even lower than in Brazil, they had to add 870 kilograms of nitrogen. This is 2 tons of urea to have the same yield of Brazil by adding in Brazil just fresh cells, 100 ml of fresh elite strains of soybean. Therefore, we are saving emission of greenhouse gas, money, and avoiding water pollution. But then we have, because in Brazil, one important thing is that we do not have monocrops anymore. We crop at least 2 crops per year, sometimes 3 crops per year. And then the farmers said, well, what do you have to offer to me after the soybean? So we went to our cultural collection and looked for strains that could be helpful for non-legume plants such as maize and wheat. And we found elite strains of Azospirillum brasiliense, which main features are a little bit of nitrogen fixation and a huge promotion of phytohormones, especially indole-acetic acid, that increase a lot root growth. And the first inoculant was released for these grasses in 2009 and 10. And it was amazing. We did not know what to expect. In just 10 years, we saw a huge response from the research. We found more than 100 field trials published in papers with results, and then we did a meta-analysis, and we confirmed statistically significant increases in grain yield of 5.4%, and very important, of 12% in root growth. More root growth is more uptake of water, of nutrients, and then taking up more water, we saw more tolerance to water stress. We also have sugarcane. It's a very important crop in Brazil, and we have an inoculant with Nitrospirillum amazonense that's an endophyte of sugarcane, so has a higher capacity than these that are in the rhizosphere, such as those of maize and wheat. And this Nitrospirillum amazonense can supply almost 50% of all nitrogen needed by the sugarcane. Next, and for the rice, high productive rice that we grow in the south of the country, we saw that Azospirillum brasilensis could reduce 25% of the nitrogen. Next, very important is this new approach that we have, that we are putting lots of efforts in this new view of microorganisms, increasing nutrient use efficiency, because we are short of fertilizers, they have environmental problems, they affect soil biodiversity, so we want to take advantage and use them as much as possible. And then microbes that improve root growth are a major thing in our research now. And for you to have an idea here, we did this experiment with N15, so nitrogen labeled. We supplied maize with 100 kilograms of nitrogen. And then the control plant, without the bacteria, took 34% of this nitrogen, but by adding the bacteria, root was bigger, and then 58% of the nitrogen was taken by the plant. So an increase of 70% in the use efficiency of the nitrogen fertilizer. And then we have a beautiful, robust set of data that nobody has in the world. So we had a decade of studies, 30 field trials. And then we were able to prove, then we talked to the farmers this last year, that in every experiment, 100% of nitrogen without inoculation was statistically similar in grain yield than 70% of nitrogen with the bacteria. Therefore, we went to the farmers when we said that they could save 25% of the nitrogen fertilizer that they were applying by using the microbial inoculants. And this was a level of 9 kilograms of nitrogen per hectare. That's the usual doses in Brazil. If we have higher doses like 150, more similar to what you use in Europe, you can save much more. And then we decided to combine microbial process because we were thinking about rhizobia for the legumes and other bacteria for the non-legumes. But then we thought, well, they have different microbial process. What if we join the industry of nitrogen fertilizer, that's the rhizobia, with the industry of phytohormones, that's Azospirillum brasilensis? Then we saw that increase that I said of 8% in soybean grain production, when we add just the rhizobia every year, we double the benefits. Then this was what we called co-inoculation of 2 strains in the soy, released in 2013-14, and amazing, again, we saw a huge response for the research, more than 50 publications with field experiments, and doing a meta-analysis of the results from these publications, we confirmed statistically significant increases in all parameters. And our farmers, they tried, and they are increasing every year the use of this co-inoculation, such that in this short period of time, it's already 35% of the whole area crop to soybean in Brazil. I'm talking about 70 million hectares, okay? So it's a huge adoption by the farmers. And microbes today we know, and we are investigating because they can help agriculture in many other ways than plant nutrition. Like the legumes, we are measuring, and we see that not only they are capable of supplying all the nitrogen needed by the plants, but also they enrich the soil with nitrogen. For example, soybean, we are measuring enrichments of 30 to 60 kilograms of nitrogen per hectare. And now towards our major problem for the following years, this water stress, we had also an inoculant with a bacteria that was isolated from our semi-arid region, that has several mechanisms of protecting the roots against loss of water. And so we have also this first inoculant to help to mitigate water stress. And of course, we do have problems. As Glauco mentioned, our major problem in Brazil is the area with pastures. That's about 2.7 times all the area with all the other crops here. 60 to 70% are degraded. So this is our black thing in the Brazilian agriculture, but Glauco explained that we have now a program to recover, and we saw that microbes can help a lot with that. We released an inoculant in 2021 that after 10 years of research, and it was amazing, we saw that our major problems in these areas are nitrogen and phosphorus, so we combined bacteria for nitrogen and phosphorus, and we saw that this combination of bacteria allowed an increase in biomass production of 22%, an average increase, and also the contents of nitrogen, phosphorus, and potassium. So the cattle would have not only more food, but food of better quality. In addition to all environmental benefits, like it was equivalent to an application of 40 kilograms of nitrogen per hectare. The roots are just incredibly higher, so a sequestration of almost 200 kilograms per hectare of carbon and lower pollution of water reservoirs. Of course, Brazil above ground biodiversity is well known. It's estimated that we have 50 to 20% of the whole biodiversity in the world. Although we do not know, but we think that below ground is the same. And here one example from our culture collection at the EMBRAPA Soybean Center, we started to bioprospect, and we saw that we have bacteria and fungi too that can solve all the problems that we may think of in agriculture in the next year. So there is a huge potential for microbiological products. But we can never stop studying and doing research. So if we had the Green Revolution in the 1970s, that Norman Borlaug received the Nobel Prize, we believe that now we are in another revolution. We are in what we are calling the green revolution. That's the use of bioinputs, microbial bioinputs in Brazilian agriculture. If you see here, the market is really very, very impressive today. So doses of inoculant, we had an increase of 74% of the commercialization in the last 3 years. The value of the biocontrol market is also increasing a lot. And the number of industrial industries producing these biological products has also increased more than 100% in 5 years. So we are in Brazil in this micro-green revolution, increasing a lot the use of microbial bioinputs. What helps a lot in that is that we have 70 years of extension in Brazil telling to the farmers about the benefits of microbial bioinput. So today we have the largest world adoption of inoculants by farmers. With 85% of the farmers that grow soybean, they use inoculants every year. This is 37 million hectares. And here it's not me that say, it's a group from Australia. Due to this use and due to the research searching for elite strains, we are the leader in the world. We have the highest BNF contribution due to the soybean, mainly due to the soybean nitrogen fixation. And so it's important. We do research, we believe, but we go to the farmers, explain to the farmers, they try, they test, and they use. And here in a short period, okay, because with grasses we just have 10 years of the release of the first inoculant, but look, just 10 years of extension, we have already for the maize crop, 1/4 of the area cropped in the summer crop using inoculant, and almost 50% of the area cropped to maize in the winter crop. And what I love about microbial technologies is that they are for all types of agriculture and farm. And I have here a situation that happened to me that represents very well this. It was May 2018, the same day, the same message. My colleague, Marco Antonio Nogueira, was in an internal seminar of one of the biggest agronomic groups that we have in Brazil. They were cropping only 670,000 hectares. Today they are already with 1 million hectares. And I was in this project in Africa of the Bill and Melinda Gates Foundation that attended 550,000 farmers with a maximum of 1 hectare. And we both were delivering the same message, using the same strains, and the contribution was exactly the same for the large farm and for the small farm. And that's the beauty about the microbial technologies. They are for all. They are for soil health and planet health too. To finish, I would like to give just some numbers because maybe you do not have an idea about the importance, what the microbial inoculants represent for the Brazilian economy and agriculture. So we today, as I said, we have this leadership of biological nitrogen fixation. And if we consider what we produce, the area cropped to soybean, the average yield in the last crop season, and the price of urea at the Brazilian market, we have the incredible figure that Brazil saved $25 billion last year, but not by not using nitrogen fertilizer with the soybean. This is $800 per second due to this wonderful bacteria fixing nitrogen. And as important as this economy is that we did not release to the environment 240 million tons of CO2 equivalent. Therefore, I hope that I convinced you of the importance and of the potential of the microbial bioinputs in Brazil, and to say that these benefits are for everybody, because they mean the use of microbial bioinputs. It means for the plant greater sustainability, for the farmer economic benefit and quality of the life, and for all countries also economy. Thank you very much.